Hi guys, welcome to Unit 2, Chapter 3, Cell Signaling. We are still talking about the plasma membrane or the cellular membrane, but now we're talking about it in the context of sending and receiving signals. As always, if you want to click on this book, you are welcome to use the guided notes and turn those in for extra credit after the exam. So there's a good opportunity to raise your score a little bit. Let's just dive in. We are going to talk about cell-to-cell communication. There are some universal truths to all communication via cell-to-cell, but there are some very specific examples that we're going to go over. So we're going to talk about it in a general term, in the universal mechanisms, but we're also going to talk about specific pathways. Yeast, for example, these are our yeast examples. We have A yeast and alpha yeast. You can see that alpha yeast is producing this red square ligand or cellular molecule, and the A yeast is producing this blue round signaling molecule or ligand. Now, yeast normally reproduce asexually, meaning that they just bud or produce a clone of themselves, but occasionally, if their little signals match up perfectly, they may combine, creating an A-alpha complex, sharing genetics, and able to make a more genetic variable offspring, so healthier, more genetically variable offspring. This only happens if the receptor on the A side perfectly matches the signal on the alpha side, or if the receptor on the alpha side perfectly matches the signal from the A side, which you can see here is what has happened. And this signal has caused elongation of the yeast molecules towards the direction of the receptor, and eventually they combine. So this is an example of cell-to-cell communication. Now, they were only able to respond to that signal because they had a specific receptor. If they did not have this specific receptor, they would not respond to the signal. It doesn't matter how many signaling ligands or signaling molecules are sent here. If the receptor does not match, it will not respond. So that's an example of a regulator. The transduction pathway is just anything that happens after the signal is received. Once the signal is received, a cascading signal is kind of sent inside of the cell, and the cell responds to it after it has that specific receptor. So it does have to have a specific receptor. Local regulators are chemical signals that influence cells that are very close by. For instance, growth factors. If a cell is very happy, it's doing well, it will send out a growth factor, and itself as well as those cells around it will grow bigger because of that growth factor. The same thing with cell death. If the cell is not doing very well, and it's struggling or it has damaged DNA, it will send out a signal to cause itself to die. We'll talk about that a little bit. Neurotransmitters are a type of local signal, and we'll go over those. More long-distance signaling includes things like hormones. Okay, we don't really go over pheromones. Pheromones are outside of the body. We're only going to be talking about hormones, which are signals that are sent from one organ of the body to another through the bloodstream. Both plants and animals have hormones. So plants do have hormones. And the ability for those cells to respond to the hormone requires that specific receptor. So the signal will not be received unless it has a specific receptor. There are four main categories of signaling. The first type of signaling category is autocrine. We see that word auto. Auto means self. So it's going to produce a signal or produce a ligand, send it out into the cytoplasm, and then it will also have a receptor that receives that same ligand. So a lot of autocrine signals can also be for cells around it, because we're sending that ligand out into the cytoplasm. But for the most part, autocrine is going to be cells targeting themselves. For instance, growth factor. If the cell is doing really well, it'll produce a growth factor, which will then be received by its own receptors, telling it to increase production and increase in size. Cell death also occurs this way. So those are both good examples of autocrine signaling. Then I want to talk about direct signaling and paracrine signaling. They're both very similar because it's between two adjacent cells, or two cells that are right next to each other. But you can see that the direct signaling crosses a gap junction. Gap junction is a connection between the two cells. So it allows communication between the two cells without that ligand possibly getting lost in the cytoplasm. The ligand goes directly to the next cell and never has to leave a cell. In plants do a similar thing, but they use something called plasmodesmata. So direct signaling can happen through a gap junction or a plasmodesmata. Paracrine signaling, however, para means nearby. So the cell will release a ligand or a signaling molecule into the cytoplasm through this gap. And then the target cell has to have a specific receptor for that ligand or that signaling molecule, or it will not respond. But if it has the receptor, it will respond. And the best example of paracrine signaling is going to be neurotransmitters. Your neurons produce specific ligands or specific neurotransmitters that travel across this gap. And this adds an extra level of control because the cells can control how much gets into this gap, and it can sustain the signal for longer if the ligand remains inside of this gap. So there's a lot of reason for paracrine signaling. The last kind of signaling that I want to talk about is called endocrine signaling. Endo meaning inside. Don't get that confused with autocrine. Endocrine means that it's going to be inside of your body or inside of your bloodstream. In this case, the signaling cell will produce a signaling molecule or a hormone. Okay, we're going to call them hormones now because they're traveling through the bloodstream. This will theoretically affect all the cells in your body, except that not all of the cells in your body respond because only the target cells will have the specific receptor to be able to receive that signal molecule. So we'll also talk about hormones and how they travel through the body. Now, I've used this word ligand quite a bit. Ligand means a molecule that specifically binds to a receptor. This is the signaling molecule. Now, ligands have lots of different characteristics. No one thing is a ligand. A ligand could be considered a hormone, and that could be a lipid-based hormone like a steroid, or it could be a protein-based hormone such as insulin. Depending on the specific characteristics of the ligand will determine what kind of receptor will receive that ligand. For example, this ligand or this signaling molecule cannot cross the phospholipid bilayer directly. Since it can't cross directly, it will respond to, or the cell will collect it through a surface receptor protein. So since the ligand can't travel into the cell itself, it's stuck on the outside of the cell. Ligands like this are mostly polar or very large. They are water soluble, so they're fine in the cytoplasm, but they cannot cross the membrane directly. So these are very large or very polar ligands. Whereas small non-polar ligands such as testosterone, which is a lipid-based or a steroid-based hormone, can cross directly without any issues. Since it can cross directly, we find these hormones free in the cytoplasm. And so the receptor for this hormone is going to be inside the cytoplasm, or a cytoplasmic receptor. Okay, this is only for very small lipid-based hormones or steroid hormones. Protein hormones cannot cross the phospholipid bilayer, so they're going to be using surface receptors. But steroid hormones are going to be using cytoplasmic receptors since they can diffuse directly across the membrane. So the prefix endo means inside. So with that logic, endocrine must mean that it's a signal sent from a cell to itself. Is that true or false? Well, that is false, because if you remember, endocrine signaling is signaling sent through the bloodstream, so it's long distance. Autocrine is when a signal is sent from a cell to itself. What type of signaling would be found in the synapse of a neuron? Well, this is an example of a neuron. Neurons have these long axons leading to an axon terminal, which just barely doesn't touch the next cell. So we have something called the synaptic cleft, or the synaptic gap. It's this distance where these cells are just not quite touching. Now, the presynaptic neuron that's sending a signal will produce neurotransmitters, or ligands. So this is a ligand, a neurotransmitter. And it will send those ligands into the gap here when it's ready to send a signal. The cellular response is determined by how many receptors are on the cell surface of the receiving signal. Once we have the receiving signal, then a cellular response can happen. Now, we can control this by sending more neurotransmitters into this gap, or increasing and decreasing the number of receptors. So there's lots of regulation here. Also, if we want to remove this signal, there is a reuptake transporter that will recycle these recycle these neurotransmitters into vesicles. So to end the signal, we will reuptake these neurotransmitters, removing the ligand from the receptor, which will end the signal. So what type of signaling is this? This is paracrine signaling. Paracrine signaling across a gap through the cytoplasm. All right, which letter represents a ligand? Well, A. A is the only one that could represent a ligand, and it matches perfectly to the receptor at B. Now, this ligand looks like it cannot cross the phospholipid bilayer, so it's bound to a receptor on the cell surface. Some ligands, however, can cross because they are lipids or steroids. All right, let's talk about the 3 stages of cell signaling. This is a very general diagram. This is not a specific diagram, it's a general diagram. But all signals follow this pattern. First is reception. When the ligand binds with the receptor, creating a ligand-receptor complex, this is called reception. The signal is received. And the signal can only be received if the specific receptor matches the specific ligand. This can happen on the cell surface, or it can happen on the inside here if the ligand is lipid soluble and able to cross the phospholipid bilayer. So this would really only happen with steroids. Step 2 is transduction, when the signal is being sent through the cytoplasm. So anything happening here is called signal transduction. And this part can get pretty complicated, but it's kind of like a set of dominoes where the receptor will send a signal to the next molecule, which will send a signal to the next molecule, which will send a signal to the next molecule. As this domino effect happens, we call it transduction. We're transducing the pathway, okay, the transduction pathway. The last part, or part 3, is going to be the cellular response. Now, this is very open-ended. The cellular response could be literally anything. This could send a signal to the nucleus to start transcribing and translating a specific gene, producing a specific enzyme or protein, which could even be another receptor or even another ligand. It doesn't matter. It could be anything. It could also be turning on or off a particular cellular response. So it just depends. This is very, very open-ended. But response is some sort of activation of a cellular process. All right, This says the transduction step. What is the transduction step? So I have step 1, step 2, and step 3. I know that step 1 is reception. It's reception because we are receiving the signal into the cell. We know that step 3 is the response because we are translating a specific gene here in the nucleus. This represents the nucleus. Now, anything happening between the receptor and the response is going to be transduction. So this is the transduction stage, and this image here is showing a transduction cascade or a signal cascade where we have that domino effect. One molecule signals the next molecule, signals the next molecule, until eventually it gets to the nucleus. All right, this one is asking which letter is the receptor. This image should look familiar because it's the same one we used earlier. We already learned that this is the ligand, so that's not possible. And all of this has to do with the signal cascade or the transduction. B is the receptor molecule, and it will only respond if it is activated by its specific ligand. So which letter represents the receptor? B. Here it says drag the icon to the reception step. Okay, we don't have an icon, but we can identify it. Here we have a ligand, and it looks like it is a steroid ligand or a lipid ligand because it can cross the phospholipid bilayer. Here we have the cellular receptor that binds perfectly with it. And then here is where we have the ligand receptor complex. This is the reception stage. So this one right here, and notice that it is inside of the cytoplasm. It's not on the cell surface. So reception can happen inside the cytoplasm only if the ligand can cross the phospholipid bilayer. So it has to be very small and it has to be lipid soluble. So a steroid is perfect for that. And here we see it activating some sort of transcription and translation of a specific gene. Here we see it creating a protein of some sort. All right, response. Which one is the response state? Well, this is interesting because this is actually an IP3 cascade where we have reception right here. The ligand is joined with the receptor, and it creates a signaling cascade that goes this direction, okay? And this is actually a molecule called IP3, and its job is actually to activate this other second messenger. This is called a second messenger. Okay, in this case, this is the endoplasmic reticulum, the ER. And if you remember when we were learning about the ER, one of the jobs of the endoplasmic reticulum is to house a bunch of ions. So in this case, it is calcium. And the IP3 causes this receptor, which is a ion receptor, to release a bunch of calcium, and that leads to some sort of cellular process. In the muscles, it may be muscle contraction, but we're releasing a bunch of ions which will increase the positivity inside the cytoplasm. It will cause the cytoplasm to become more positive since calcium is electropositive, leading to some sort of response. So this is the response in this image. All right, we are going to talk about 3 different kinds of receptors. The first receptor that we're going to go over is ion channeled link, similar to this one right here. So ion channels are receptors that will open up an ion. If you remember last week, we talked about a concentration gradient. And here in this image, we can see that we have a very large concentration of ions on one side of the cytoplasm or one side of the plasma membrane. When the ligand matches this receptor, it allows the receptor to open a channel, and all of those ions will rush into the cell, okay? So in this case, it looks like it's going into the cell. And this is called a gated ion channel, okay? Gated ion channel. It's gated. It's closed most of the time unless the ligand binds, and then it opens, allowing a flood of ions through. To end that signal, all we have to do is release the ligand, and then the gate closes again. So this can be used over and over and over again as long as there's a concentration gradient. So what I wanted to cover in this video is the ion-gated ligand channel. Okay, so here's the ligand, which allows the ions to flow through the gate. Here we can see that again. We have a bunch of ions on this side of the cell. When the ligand binds, the gate opens, allowing the ions to flow into the cell. When we want to end this signal, the ligand will be released. All right, so we have covered ion-gated channels. Now we're going to talk about enzyme-linked receptors. So one of the types of receptors that we're going to talk about is something called tyrosine kinase. Okay, so that is a big word, but we will see it over and over and over again, and there are lots of test questions about tyrosine kinase. So the general pattern of tyrosine kinase is that we have these two tyrosines. They are receptors. They do span the phospholipid bilayer, so we have the signaling molecule on the outside. Both of these receptors have to be activated for this to work. Once the tyrosine kinases are activated by a ligand, they create what's called a dimer. Okay, that just means that we have two molecules together. We can have a trimer or a dimer. Trimer is 3, dimer is 2. A dimer is tyrosine kinase. So if you ever see tyrosine kinase, we're going to be talking about a dimer. And you notice that tyrosine kinase has 6 different tyrosines, or 3 tyrosines on each kinase. Okay, these are kinase proteins. Those 6 tyrosines become very, very important because they can strip a phosphate off of ATP. So 6 ATP are utilized to get 6 different little phosphates. 1, 2, 3, 4, 5, 6 phosphates, leading to 6 ADP, just meaning that we have 1 less phosphate. So these will go back to be recycled later. But this activated ATP is utilized, and we phosphorylate the dimer. Phosphorylation means to add phosphates. So we have phosphorylated the dimer. That's not where the signal ends, though. We're using these phosphates. We're activating something called a relay protein, which means that a tyrosine kinase can activate multiple cellular responses. And we will phosphorylate these relay proteins with this new phosphate from the tyrosine. So the tyrosine will strip the phosphate from the ATP and give it to these relay proteins. Once the relay proteins are activated with the phosphate, they can go perform a cellular response. So I'm not very specific about the response here because they can do multiple responses with one signal. So one single signal, which means a dimer, can send multiple cellular responses because of this phosphorylation. So anytime you see a question about 6-phosphates, we're talking about tyrosine kinase proteins. When they are inactive, they are monomers. When they are active, they are a dimer or a pair. Now, to end this signal, the phosphate is used up by these relay proteins, and once the phosphates are used up, it will end this signal, releasing these ligands. right. So here we can see the example of tyrosine kinase again. We have the inactive tyrosine kinase monomers and then the signaling molecule that will bind to the receptor. Both have to be activated at the same time to create a dimer. Once it's dimerized, it will strip phosphates from ATP, producing ADP, up to 6 different phosphates. Once we are activated and we're completely phosphorylated, we have all those phosphates, it can activate any number of cellular responses using relay proteins. So these relay proteins will grab onto that phosphate, become active, and do some sort of cellular response. So now we have talked about enzyme-linked receptors. Let's talk about the last kind, G protein coupled receptors. So a G protein coupled receptor, or GPCR for short, there is a question asking you about that. So if you see GCPR, we're talking about G protein coupled receptors. These are probably the more complicated receptors because they have a signaling cascade. It is also considered a multi-pass protein receptor, meaning that it passes through the membrane 1, 2, 3, 4, 5, 6, 7 different times. So it passes through the membrane 7 times, making it a multi-pass kind of receptor. But it is a surface receptor, meaning that the ligand does not have to cross the phospholipid bilayer. It's going to receive the signal from outside in the extracellular fluid. It is attached to something called a G protein. Yay, here is the name, G protein. The G protein is actually a peripheral protein, an internal protein that does not cross the phospholipid bilayer. So the receptor will cross the phospholipid bilayer, but the G protein will not. And the G protein is actually made out of 3 different pieces. We don't need to know that for now. But once it becomes activated, this GDP, which is similar to ADP, gains a phosphate. Once it gains a phosphate, it is GTP, or similar to ATP. Very, very similar, still has 3 phosphates here, and the phosphate is the important part. It's just G instead of A. So when the ligand binds to the receptor, this G protein becomes activated with GTP, and then it will travel along the membrane until it bumps into a membrane-bound enzyme, this enzyme right here. Now, it phosphorylates that enzyme that then activates a second messenger, which then can activate a third messenger, so on and so forth, leading to a signaling cascade. This is the most common type of G protein coupled receptor, where the G protein disassociates from the receptor once it becomes activated and sends a signal to an enzyme that will then start the cascade. Occasionally, it will disassociate, and another piece of it, maybe the alpha or the beta complex of the G protein, will go and do some other type of cellular response, because this is actually made up of 2 major parts and a minor part. So it can do multiple things, but the most common one that I talk about is going to be the enzyme process. So G protein coupled receptor has an external receptor, meaning that this ligand cannot cross the phospholipid bilayer. It could be insulin, for example, that does not cross the phospholipid bilayer. Once it binds, it activates the G protein, which then disassociates into different pieces. So one piece might go this direction, another piece might go this direction, with the assistance of GTP. GTP, easy to remember because it's the G protein. It uses GTP instead of ATP, which then will activate an enzyme, which starts a cascade of second messengers. Okay, this leads to a general cellular response, which again can be literally anything, part of the cellular response. All right, so like I mentioned, there are multiple different types of G protein coupled receptors, but they're all going to use GTP to activate this messenger right here. This comes in two main pieces, the alpha and the beta. Gamma is also on there. And they can activate a membrane-bound enzyme, which will then lead to a signal cascade. To end the signal or to cease the signal, we remove the ligand. But this happens in lots of different parts of your body. For instance, norepinephrine causes a signaling cascade that involves cAMP or a protein kinase. Glutamate will involve an IP3 or a calcium release. Okay, dopamine can signal also a cAMP or a protein kinase signal. So we have lots of different types of G proteins here. All of these second messengers are going to involve phosphorylation or removing that last phosphate from ATP, or in this case, GTP. But the signaling cascade involves something called pyrophosphate. Okay, so phosphodiesterase is going to be what converts cAMP or CAMP into AMP. So if we're talking about the norepinephrine or the dopamine signal, this has to use phosphodiesterase for its signaling. Okay, so we have to use phosphodiesterase for our second messenger. Okay, second messenger is just what is happening after the reception of the signal and before the result. Okay, so anything in here is in the transduction pathway. Those are second messengers. So in this case, the second messenger is protein kinase, and in this case, the second messenger is IP3. But that uses phosphodiesterase to convert cAMP into AMP. This is a bigger picture of these receptor cascades. So this is step 1, or the signal reception. Okay, step 1. This is signal transduction, step 2. And this is cellular response, which is step 3. And again, anything along this signaling cascade, which is like a case of dominoes, we're going to use phosphates from ATP, or in this case, GTP. Down here, we're using ATP to phosphorylate the next one in the line. So what we have is a series of enzymes that are being phosphorylated. In this case, cAMP is turned into AMP using phosphodiesterase. In this case, IP3 binds to the endoplasmic reticulum, causing a large release of calcium. So both are different versions of a G-coupled protein receptor. So we've talked about G protein receptors. Let's look at this. It says drag the icon to the second messenger step. So the second messenger. Well, I know this is signal reception, and I know that this is cellular response. So that's step 1 and step 3. Everything in here is going to be signal transduction, or step 2. So this would be our second messenger. It's second in line. We have a third messenger, a fourth messenger, just a series of second messengers. And this represents phosphorylation because we have that energy. So each successive enzyme is phosphorylating the next enzyme. So this is a signaling cascade, a cascade. A signaling cascade is always going to happen in the transduction step. All right, this is saying what kind of signal this is. What I see is a ligand that opens a gate, allowing a whole bunch of ions to flood in. So we're going to call this an ion-gated channel. It's a ligand ion channel. All right, which of these represents the G complex? Well, it's the one connected to GTP. When it's inactivated, it would be over here connected to the receptor, but once it becomes activated, it phosphorylates, creating GTP, moving on over to activate this enzyme complex, which then will phosphorylate our second messenger. So here's our second messenger here, and now we're using ATP to phosphorylate it. All right, which one of these represents the activation enzyme? Right here is the enzyme being activated, letter C, because we're activating it with phosphate from ATP. Actually, we're activating it with a phosphate from GTP. I apologize, from GTP. And this is one of the major steps in a G-coupled protein receptor. So this is our enzyme being activated. This is also an enzyme being activated, but it's part of our signal transduction. All right, transduction isn't a linear event. It doesn't happen in a straight line. Several things can happen. We can amplify the signal, or we can combine the signal. So for instance, looking at this image, we have several different receptors that lead to the same second messenger, which means that if we have a ligand that doesn't quite match this receptor, maybe it matches this receptor and it can still activate that pathway. Even better if we have multiple signals that activate the same pathway. This is a type of confirmation. Okay, we confirm this pathway, we make sure that we're going down the correct pathway. If we have another type of signal that leads to a different second messenger, maybe we need both of those signals to get to our third messenger to lead to our cellular output. So this is kind of a checks and balances as part of the phosphorylation cascade. These relay molecules or this phosphorylation cascade can branch, or, in this case, it's kind of a funnel. It's taking multiple signals and leading to one cellular response. And we need multiple signals to make sure that that cellular response is appropriate. The other thing that can happen is the opposite of that. Maybe we only have one ligand, but we need multiple things to happen. Instead of integrating the signal like you saw here, this is signal integration, now we see signal amplification. So in this image, we have one receptor activating multiple G proteins, and those G proteins lead to different events, different cellular events happening in the cell. But in this case, we only have one ligand and one receptor. And that one receptor leads to 3 different responses, which branches out each one of those into 3 different responses, which each one of those is 2 responses. So in this case, by the time we get to step 6 in the cellular response, we don't just have one response, we have an amplified response. So we're producing lots and lots of signal here. So let's say, for example, the response to this ligand is to produce protein A. Instead of one ligand producing one protein, which would not be very efficient, otherwise I wouldn't really need a signal, I would just produce the protein. I'm sending a signal that I don't want to just produce one protein A, I want to produce 20 protein A's. So in this case, the signal is amplified and made larger. So here we see that same thing. We only have one cell receptor, but it is activating multiple C-amps or cyclic AMPs that then will produce many, many different target proteins. So this gets big very, very quickly. All right. Looking at this diagram, where would amplification occur? Well, it doesn't occur at the reception stage, and it doesn't really occur at the response stage. It happens during signal transduction, where instead of this domino effect only affecting one second messenger, it would affect multiple second messengers at a time, leading to amplification of the signal very quickly. All right, which one of these represents amplification? This is linear and this is linear, so I know neither of those are the correct answer. This one looks like signal integration because we have two receptors that lead to one response. But here we have one receptor that leads to two responses, so I would call this one amplification. Which of these represents signal integration? Well, we just talked about that. These are linear, so it's not it. And we called this one amplification, so that can't be it. So this one is integration. Let's talk about something called apoptosis. Apoptosis is another way of saying cell suicide. It is a type of autocrine signal or self-signal, and it can also affect the cells around this cell. So autocrine and paracrine at the same time. If something is wrong with the cell, maybe it doesn't have enough nutrients, maybe it doesn't have enough space, or maybe the DNA is damaged, the cell is protecting your body from reproducing or replicating damaged DNA. Since we don't want to replicate the damaged DNA, the cell will send a signal to itself to initiate apoptosis or cell suicide. And the cell will essentially digest itself into these small vesicles until another cell will come by and undergo phagocytosis, recycling all of these components. This protects your body from replicating DNA. And if a cell does not undergo apoptosis and it does reproduce using bad DNA, it can become cancerous. So apoptosis is cell suicide, protects your cell from damaged DNA, protects your body from cancer. It's very interesting, and it is usually initiated from inside of the cell itself. So this is a type of autocrine signaling. Autocrine signaling. All right, so in that example, we were talking about apoptosis, and your cells can become cancerous if they're reproducing damaged DNA. So apoptosis helps to protect your skin and protect your cells from doing that. In this image, what we see is a G protein that instead of activating an enzyme, it's creating this IP3. So IP3 is going to then connect to the endoplasmic reticulum right here, that releases calcium. So in this case, calcium is our second messenger instead of a signaling cascade. We're still talking about G-coupled proteins though. It's just a unique pathway. We're still using GTP to activate the G protein, but now we're going to be producing this thing called IP3, which will release calcium from the endoplasmic reticulum, leading to the cellular response. So this is just an example. To look at other examples, we can go to this website and look at different types of cell signals. So you can explore this webpage if you would like to, and look at how different types of cells respond to different stimuli. So maybe a growth factor. So growth factors should cause it to grow larger and replicate. So different types of signals will affect different cells due to their specific type of receptor. So this is actually a type of paracrine signaling, where a signal is sent across a synaptic cleft in response to sunlight or any kind of light. Don't forget to print your chapter laboratory write-up. The laboratory write-up needs to be either filled out by watching the QR code or by bringing it to class. You can always do the practice quizzes and the optional homework. If you ever want me to check the optional homework, I can. Also, your SI leader should have all of the answers to the optional chapter homework, which you get to turn in for extra credit. Don't forget, you also have these extra resources, including the textbook and the flashcards. Thank you for joining me today, and I hope that y'all have a great week, and I look forward to seeing you next week.